Hi everyone, my name is Kelsey Yetsko and I'm a Master of Science graduate from the University of North Florida. And I'm going to be talking about if a threatened coral can potentially adapt to increasing temperatures. If you have any questions after the video, please feel free to contact me via my email address or my Twitter handle listed below. The species that is the focus of my talk is the Caribbean staghorn coral Acropora cervicornis, which is listed as threatened by the Endangered Species Act in the United States. This species, along with a related species, the elkhorn coral, once dominated Caribbean reefs in the Florida Reef Tract. However, a severe disease event in the 1980s resulted in major mortality, and now the species is less than 3% of its former abundance. In addition, continued summer thermal bleaching events are hampering the restoration efforts for the species in the Florida Keys and the Caribbean. Now, what is coral bleaching exactly? A coral is a colonial animal with tissue over a calcium carbonate skeleton, and within the gastrodermal cells of the coral are single-celled symbiotic algae. The algae photosynthesize and provide the coral host with 90% of its required nutrients. However, when a coral is exposed to any sort of environmental stress, in this case temperature, it can result in a breakdown of the symbiotic relationship, and the algae will leave the coral cells, causing the coral cell to lose its color and giving it that characteristic bleached appearance. While coral can regain their symbionts after a bleaching event, if the stress is too severe or lasts too long, then the coral starves without the nutrients provided by the symbiont and dies. Because of this, it's really important to understand how corals will be able to respond to and survive higher temperature in the future. To understand whether a species can adapt to climate change, we need estimates of the amount of genetic variation, or heritability, that is present for the trait of thermal tolerance. Now, before I go any further into what that is, just a quick genetics recap, since I'll be using these terms frequently throughout. When we say phenotype, we're talking about the set of observable characteristics of an individual. In this case, for phenotype, we're talking about the trait of thermal tolerance, so surviving a thermal stress with minimal to no bleaching. On the other hand, when we say genotype, we're talking about the genetic makeup of an individual. So what in the DNA of a coral that is resulting in higher thermal tolerance. Now that we've got that out of the way, I'm going to go back to this term heritability. What is it and why is it important to know? Heritability is the measure of the amount of genetic variation within a phenotype. So heritability in a broad sense is the proportion of the total variation in a phenotype that has a genetic component. And this is calculated by the equation Vg over Vp. This can also be illustrated by the graph below. As individuals go from being less genetically similar on the left-hand side to more genetically similar on the right, their phenotypic similarity increases. So on the left, two corals that aren't related are going to have different thermal tolerances or bleaching susceptibility, while on the right, two corals that are more closely related are going to have exactly the same tolerance. That means the trait in question is highly heritable, and most of the variance in phenotype has a genetic component. Now that was a very oversimplified example. However, genetic variance isn't all that simple. It's actually made up of three different components, where VD is the component that is due to dominance effects between alleles. VI is the component that is due to interactions between multiple loci or multiple genes, and this is called epistasis. And VA is the component that is additive and is actually what makes parents and offspring look alike. So broad sense heritability is all of these different components combined together, but selection only acts on the additive component, VA. So that is what we are most interested in. And that proportion of phenotypic variance that is due to these additive effects can be calculated by taking VA over VP. So once we figure out how much phenotypic and genotypic variation there is for thermal tolerance in the species, the question then becomes what exactly is influencing the higher tolerance of certain individuals? Is it symbiont identity or microbiome identity, which has been shown to play a role in many species, particularly with clade D symbionts? Is it the expression of certain genes by the coral host or symbionts? Is it the concentration of certain proteins or the activity of certain enzymes? Or is it related to phenotypic plasticity or changes in epigenetic state following a prior exposure to temperature stress? Once we know what is actually influencing higher tolerance, we could potentially develop tests to identify more tolerant colonies to use in future coral restoration work. For the purpose of this talk, I'm going to focus on the role of coral host gene expression on thermal tolerance. But if interested in other results such as symbiont identity and protein and enzyme concentration, please keep an eye out for our peer-reviewed paper, which has just been accepted in Marine Ecology Progress Series, or contact me for any questions. For this study, the main questions were, number one, how much total genetic variation is there for the trait of thermal tolerance in a nursery population of Acropora cervicornis, and how much of that variation is due to specifically additive genetic effects that selection can act on? And number two, are there differences in gene expression between tolerant and susceptible colonies? And if so, would any of these genes be good candidates for biomarkers of thermal tolerance? To start, we first obtained coral fragments from 20 genetically different coral colonies from the Coral Restoration Foundation, or CRF's Tavernier nursery. These fragments were split up as follows. We had one fragment per genotype that was stored immediately for genotyping of the coral host. Then the fragments were split among 6 tanks, 3 that were heated to 32 degrees Celsius, and 3 that were controls that were kept at an ambient temperature of 28 degrees Celsius. An example of what each tank looked like is shown here. There were 2 fragments per genotype for all 20 genotypes, although the position of the genotype was randomized between the different tanks. Here's a photo of what each tank actually looked like. Each fragment was suspended in a flow-through system, and for the heated tanks, an individual heater was placed near the source of flow. 12 hours after the heated fragments were exposed to 32 degrees, we removed 1 fragment from each pair and stored an RNA later for RNA sequencing. This RNA sequencing would give us measurements of what genes were being expressed at this specific time point, and also how much or how little those genes were being expressed in each of our samples. We also did the same thing for 1 fragment per pair in the ambient temperature tanks as well. Mortality was then measured on the remaining fragments for 26 days using a diving PAM, or pulse amplitude modulated fluorometry, which basically measures the amount of photosynthesis that is occurring by the symbionts in the coral tissue. Once values pass below a certain threshold, usually 0.2, that is correlated with severe bleaching and mortality. But we also confirm mortality by total loss of the coral tissue from each fragment. So what did we find in relation to our first question for total and additive genetic variation? First, we found that prolonged temperatures of 32 degrees Celsius had a significant negative effect on the lifespan of Acropora cervicornis. So we see on the left that the control or ambient fragment survived a total of 26 days, or a little over 600 hours, while the heated fragments all died around 20 to 21 days. However, we also found that prolonged temperatures of 32 degrees Celsius affected the different genotypes differently. There was a wide range in the lifespan among the different genotypes, with those on the far left being the most susceptible to temperature, and those on the far right being the most tolerant. The only significant differences were shown by the letters, though, with genotype M5 having a significant higher lifespan than genotypes M7 and U33. When we use this data to calculate broad sense heritability, we find that these genotypes have a high broad sense heritability. We found that capital H squared was 0.529, which means that 52.9% of the variability in thermal tolerance was due to the variation in genotypes that we saw earlier. However, when we try to calculate how much of that variation is additive, the picture is less clear. All of our different methods of calculating narrow sense heritability give us a lowercase h squared around 0, suggesting that none of this total genetic variation, that 52.9%, is additive, and therefore cannot respond to climate change. But when we look into this further, we found that there was little variation in how related these colonies were to each other, as our VR shown here was close to 0 as well. This species shows a great deal of population structure in the Florida Reef Tract, which has been shown by studies by Drury et al. And each of these colonies was originally sourced from different reef locations, meaning that all of these colonies were not really related at all, except for those that were exact clones of each other. For narrow sense heritability to be accurate, you need a wide range of relatedness. You need some individuals that aren't related, some that are distantly related, and some that are really closely related. In addition, there are empirical studies using other data sets that show in most cases, additive genetic variance makes up half or more of the total broad sense heritability. So since our relatedness estimates are inaccurate and our broad sense heritability is high at 0.5, it is likely that the additive genetic variance for thermal tolerance and cervicornis is greater than 0. We just don't know by how much. Moving on to question 2, are there differences in gene expression between tolerant and susceptible colonies, and would any of these make good biomarkers? Due to limited funds, we decided to sequence only the 3 most susceptible colonies, highlighted on the left in blue, and the 3 most tolerant colonies on the right in pink. We sequenced samples from all 6 of these genotypes from both temperature treatments, so both heated as well as control, for a total of 36 samples. What we found is that there were 129 genes that were significantly different due to thermal tolerance. When we use those genes to cluster samples based on similar gene expression, we see the samples are clearly separated by tolerance, with tolerant pink samples on the left and susceptible blue samples on the right. Samples within the same genotype also cluster closely together, showing similar gene expression among those samples for those 129 genes. And we also see that both control and heated samples clustered closely together, showing no difference. We also further filtered that 129 gene set by looking for genes that were the most consistent among different individuals that had the same tolerance. When we do this, we found that 40 of those 129 genes were highly consistent among different genotypes. And this is shown by the heat map here. So genes that are upregulated are shown in pink, and those that are downregulated are shown in blue, with brighter colors showing greater upregulation or downregulation. Susceptible samples are grouped on the bottom left and tolerant on the bottom right. The top 2/3 of this heat map is showing genes that are largely upregulated in tolerant genotypes and downregulated in susceptible, while the bottom third shows the opposite. So for example, we see genes highlighted here, like the NLR family card domain containing proteins 3 and 4, as well as the tumor necrosis factor receptor superfamily member 16, that are upregulated in tolerant genotypes, so those in the pink. And then we also see zinc transporters at 14, which is downregulated in tolerant genotypes, which we see in the blue. However, rather than look at individual genes, sometimes it's more helpful to look at groups of genes. When we do that, we find that most of the genes that were upregulated in tolerant genotypes, the pink bars, were significantly enriched for biological processes that promoted the integrity of symbiosis. So this included processes like cell adhesion, which are circled here in green. And this has been implicated before in the bleaching process with the loss of symbiont cells. We also see upregulated genes enriched for inflammatory responses, which are part of the innate immune response. And this has also been shown to be involved in managing symbiosis and symbiont recognition. For genes that were downregulated in tolerant genotypes, the blue bars on the bottom, these were mainly involved in transport-related biological processes like the ones circled here, especially those related to ion transport. However, only the top one for inorganic ion transport was significantly enriched. The downregulation of ion transport may be related to a trade-off between survival and growth, with ion transport being especially important for calcification. There is evidence that colonies with enhanced survival do have slower growth, so that would be an interesting question to follow up with these particular genotypes. In summary, for question 1, there are significant differences in thermal tolerance among cervicornis colonies from CRF. And we see that our broad sense heritability was 0.529, which means that 53% of the variance in thermal tolerance was due to differences in coral genotype. However, we're unable to make any conclusions about the level of additive variation in thermal tolerance for these colonies, although it is likely to be greater than 0. For more precise estimates of additive genetic variation, more related colonies will need to be sampled from the same reefs in order for this method to be accurate. For question number 2, we found that differences in thermal tolerance corresponded to gene expression differences for 129 significant genes, 40 of which were especially consistent among genotypes based on tolerance. Genes that were significantly upregulated in tolerant colonies may be involved in the maintenance of symbiosis to prevent bleaching, while those downregulated may be involved in a trade-off to shift energy and nutrients from growth towards survival. Finally, these genes will need to be verified across a wider sample size before attempting to use them as biomarkers for thermal tolerance. And they should also be tested under other ecological stresses to confirm that these are thermal tolerance specific responses and not just general stress responses. And with that, I would like to thank my funding sources, particularly the Patty Foundation, the American Museum of Natural History Richard Gilder Graduate School, and the University of North Florida, as well as my thesis committee, my RAU student Michelle Ross, and our collaborators at FIU for all of their help. Please feel free to reach out to me again with any questions, and thank you all for watching.